Good afternoon and welcome. I'm Patrick Cunningham, and along with Jeff Normandin, we will be your moderators today for this virtual MEGUG Brown Bag Lunch and Learn. All attendees are in listen-only mode, but if you have any questions for Megan or any of us, please use the questions panel and we will do our best to answer them. We're very excited to have Dr. Meghan May presenting on clinical and epidemiological insights into the COVID-19 pandemic from Geospatial Sciences. But first, we'd like to say a word about the main GIS users group. We need you. We hope you are all members, and we also want to know if any of you have thought about possibly running for the main GIS users group board. Our annual meeting next month will have elections for some open seats, so if you're interested, please let us know as soon as possible. The main GIS users group is a nonprofit organization that promotes awareness of and encourages the coordinated development and effective use of geographic information and related technologies. MEGUG focuses its efforts in the areas of policy liaison and networking, research, education, and public relations. The group facilitates discussion and exchange of information and ideas in these areas by hosting regular meetings, workshops, and roundtables. And these are all open to both members and non-members. As you know, this year we have pivoted to our virtual Brown Bag Lunch and Learn series, and we're very pleased that it has been well received and well attended. We are always looking for new ideas for future Brown Bag Lunch and Learn, so please share those with us. If you have a paper or presentation you'd like to present, we would be happy to hear from you and accommodate you. A word about the main GIS users group board. You can see our members here. These are the board members. Total membership is somewhere around 100, I believe, right now, but our board is comprised of folks from around the state in various GIS roles in Maine and abroad. Word on those upcoming events. So next month will be our MEGUG annual meeting, and it will be a little bit of a different format. This event should last about an hour and a half. We do try to respect that this is a lunch and learn, and everybody gets their share of Zoom and virtual meetings, so we won't make it be too long. We will have a presentation, as we have at all our Brown Bag Lunch and Learns, and that one will be from the Maine Inland Fisheries and Wildlife on the success of the eagle recovery efforts, sharing some story maps and some other information. We will also have, as it's our annual meeting, brief presentations from our gold sponsors, Esri, Quantum Spatial, and Blue Marble Geographics. And then we'll have a meeting in the meeting where we will present to you what we've been working on and talk about our future plans and ask for your input on those efforts. And then, of course, we will hold the elections. December 8th, our next Lunch and Learn then will be on high-resolution land cover and forest mapping products from Maine. We're going to have two folks from NOAA presenting. And in January, it's still TBD, so we need your ideas. As I said earlier, please submit your talk ideas to megugboard@gmail.com. We want to hear from you. As we conduct all our virtual Lunch and Learns live, we also do record them and then we post them on our YouTube channel. So you can see here that we've already got 4 up there. So if you miss anything or you have a colleague that you'd like to share this with, point them to our YouTube channel and please subscribe. I'm going to now hand it over to Jeff to introduce our speaker. Jeff?
Thanks, Patrick. Thank you, everybody, for attending today. And most of all, would like to thank our speaker today, Dr. Meghan May. Dr. May was appointed to the Department of Biomedical Sciences at University of New England College of Medicine in 2013. Before that, she was in the Department of Biological Sciences at Towson University, where she was the Fisher Endowed Chair of Biological Sciences from 2012 to 2013. She was a research assistant professor in the Department of Infectious Diseases and Pathology at the University of Florida's Emerging Pathogens Institute. And she has a BS degree in microbiology from UNH and a MS and a PhD in pathobiology and bacteriology from the University of Connecticut. Dr. May contributes pieces to local and national print, radio, and television media on infectious diseases, and has authored 48 peer-reviewed publications, 15 invited book chapters, and has given several platform presentations at national and international meetings. So with that, thank you, Dr. May, and let's go ahead with the presentation.
Hey, thank you so much, Jeff. That was for your kind introduction. I'm going to be wildly unprofessional and point out that Jeff left out the most important connection that we share, which is, of course, that our respective two children are classmates of each other, and our boys are best buddies. So
that's true. And thank you for the socially distant trampoline activities yesterday.
Anytime. It's always challenging when suddenly school is off for 2 weeks and we have to make it up as we go along. But in any event, I was very excited when Jeff asked me to give this talk, because it's one thing that I've come to appreciate is that as scientists and as engineers, we tend to talk extensively amongst ourselves, and we don't always go outside our expertises as much as we should, because I find that every time that I do that, I learn invaluable lessons, and often I come away with new skills. And one of the things I hope to impress on you is that in the area of emerging infectious diseases, it's critically important that we incorporate any and all scientific skill sets as we try to navigate what are often very much emergency situations. So with that being said, I'm going to talk a little bit at first about the virus itself and how we came to be into this situation that we're currently in. But I think in order to best appreciate that, we have to start a little bit at the beginning, which is to point out something about virus and host interactions. There are innumerable nerdy details about how any specific virus interacts with any specific host cells, but there really is one fundamental principle here, and it's best exemplified by pegs and holes, believe it or not. It's slightly more complicated than that, but not much more complicated than that. Every virus has something called a ligand or an adhesion, and every host cell has a receptor. Those two things have to be able to bind and interact with each other in order for a virus to infect a host cell. So we can think of the virus side of this as a peg and the host cells part of it as a hole. You have to have a round peg and a round hole in order for that virus to infect that cell. Again, a little more complicated than that, but that's the basic gist. And okay, sorry, I was having a strange cursor issue for a second. So if I had to be slightly more adult about it and put some biological looking diagram up, it would look something like this, right? If this along here is the surface of a host cell, if we're thinking about COVID-19, that would be, say, the epithelial cells that line your respiratory tract. And you have a virus that's floating along next to it. It has to be able to find, using its pegs, the appropriate receptor in order for this virus to get inside this cell and do what it does, replicate itself, damage the cell, and ultimately kill the cell. Okay, so what happens if you have a square peg in a round hole? Absolutely nothing, right? So if you have a virus that you inhale whose peg doesn't match the hole, nothing is going to happen. You will have no ability of that virus to enter that cell, replicate itself, and start causing damage. So you might think of as an example, if your dog has kennel cough and the dog is extremely sick and they're coughing all over you, if you inhale their pathogen load, nothing's going to happen to you. You're not going to get sick because in that instance, you've got a square peg round hole mismatch. All right. Every once in a while, though, you can have new round pegs that are going to fit into your holes perfectly well. And when that happens, you get something called a zoonotic spillover event. Virtually all new novel pathogens result from zoonotic spillover events. They were infectious agents of some animal, and they've jumped into humans, and they can now spread often from human to human to human. We have to think about this as a rare event that happens not very often against the backdrop of plenty of opportunity for it to happen. If you think about how many times you've been close to an animal, whether it's a cat, a dog, a bird in your yard, you walked by the same tree that just had squirrels running all over it. We interact with animal microbes all the time, but most of the time, absolutely nothing bad happens because we don't have the right pegs and holes. When this does happen, and we do have the right pegs and holes, we can get into a catastrophic situation like the one we are currently in. The virus that causes COVID-19 is called SARS-CoV-2. So where did it come from? We know a few things about it, and there's a lot more that we don't know, but we can extrapolate some things from it from what we know about a similar virus that jumped into humans in the early 2000s. And of course, it's got a very similar name. I'm going to talk for a second now about SARS-CoV-1, or the original SARS coronavirus, which most of us are probably old enough to remember when that happened. I had just started grad school and was working in veterinary pathology, so it was particularly interesting. So what we know about SARS-CoV-1 is that this was a virus of bats. And it doesn't seem to do anything terrible to the bats. It's just part of their normal microbiome. And what eventually we learned happened was that this virus jumped from bats into an incidental host. In this case, it was an animal called a masked palm civet cat. And what happened after that was that these civet cats were farmed for their meat, and they were sold in a wet market. This is an image of the very market where the infected civet cats were traced to. It's called the Kingpin Market, and it's in the province of Guangdong, China. So these guys are often they're often culled on site. The thought being that the meat, which is considered a delicacy, is much fresher. What ends up happening in those conditions is that you have live animals that are often in cages next to other live animals of different species, and when they're butchered on site next to other live animals, next to insects, next to produce, there's a lot of opportunity for transmission of microbes from one animal species to another. And when that happens in a very densely populated area, such as Guangdong, you have a tremendous opportunity for a lot of transmission to happen from an animal to a human. And in the case of SARS coronavirus, it so happened that this also was able to be transmitted from human to human to human to human. And then you wind up with a very large, very fast-moving epidemic. What we know from history is that with intense public health measures, this was able to be brought under control after about 9 months. So what do we know about SARS-CoV-2? Did this come from bats via cats? We don't know all of the story, but what we do know is that it almost certainly, like its close cousin, has a reservoir in bats. We don't know who this secondary incidental host was, if there even was one. It's possible that it came straight from bats to humans, though rather unlikely. We do have some candidates that are potentials. One of the first papers that we saw on this was a group that was looking at the susceptibility of different types of mammals to SARS-CoV-2. They looked at ferrets, they looked at cats, dogs, chickens, pigs, cattle, and what this paper found, long story short, was that pigs, cattle, chickens, dogs, not particularly susceptible to this. Cats, however, were quite susceptible. They got clinically sick, they shed a lot of virus, and ferrets were extremely susceptible. And this is actually starting to become much more interesting, looking at ferrets and other mustelids. There have been now 3 stories from the Netherlands of outbreaks of what looks very much like COVID-19 disease in farmed minks. The first of these, the farmers themselves were sick, and as they were caring for the animals, they spread the virus to the farmed mink, and then the mink started spreading virus from one animal to another. This happened again 3 times in the Netherlands, and this past week we had a news report of an outbreak on a mink farm in Utah, within the US. So it really is looking quite plausible that a secondary host was some kind of mustelid animal, the ferret, mink, weasel type thing. But we don't know that yet. And it's worth pointing out that this story here about the bat reservoir going to the palm civet through the market and which market it was, we didn't know the answer to this until about 18 months after the SARS outbreak or the SARS epidemic ended. So it's not unusual at all to not know this whole story right away. Okay, so what are the pegs and holes and the clinical points of both SARS-CoV-2 and its cousins, because this is potentially informative. We have 3 different beta coronaviruses that cause severe disease in humans. One causes MERS, Middle Eastern Respiratory Syndrome. Another causes SARS, Severe Acute Respiratory Syndrome, that we just mentioned a second ago. And then the third one, of course, is SARS-CoV-2, which causes COVID-19. So just to mention MERS quickly, MERS has a 35% case fatality rate. So MERS is an extremely deadly disease. It strikes most commonly people with a lot of comorbidities, which is something we see with SARS and SARS-CoV-2 as well. But an important number here is something called the R0. This is a measure of how transmissible a virus is if we take it to its most basic level. What this number means is how many people one infected person is likely to transmit to. So if you have a virus whose R0 is 1, that means for every infected person, they will be expected to infect one other person. If your R0 is lower than 1, that means you're not particularly transmissible as an infectious agent. And if it's higher than 1, you are exponentially transmissible. So MERS is extremely deadly, but it's not particularly transmissible. So that's good news. It was a zoonotic spillover event, and most of the time when you have a MERS patient, they got sick directly from an infected animal. MERS came from bats, but its secondary host that usually infects people are dromedary camels. Most of the patients have had recent contact with a camel, And when you do have a patient that transmits to another human, it's almost always from a sick patient to a caretaker because they're in such close, intense contact. The thing that we know about MERS is that its receptor, its hole, is a protein called DPP4, and you only find that in the lower respiratory tract. So while this is more deadly because it's killing cells way deep in the lungs, the silver lining to that is that it's very hard to cough virus out and transmit to another person. When we look at SARS, though, what we see is that it uses a different receptor, something called ACE2, and that's going to become important, so keep that factoid in your head. We find that it's got a much lower case fatality rate, only about 12%, which again is still pretty impressive for an infectious disease, but we see that it's a lot more transmissible, and you can take a guess at why when you look at this. So ACE2 we see is on these cells down here, but it's also in the upper respiratory tract. So while it's less likely to kill you, since it's replicating up here in the nose, in the mouth, in the sinuses, and in the trachea, you are much more likely to cough it out and infect somebody else. So SARS was, as we talked about a minute ago, it was a zoonotic spillover event, but it also spreads person to person to person. And finally, that brings us to SARS-CoV-2, which we've got a very similar situation to SARS, right? In that we have replication in the upper and lower respiratory tract. We use the same receptor that was figured out very quickly. And what we also see, though, is its R0 is actually even higher. And this is probably a reflection of the fact that it's got this lower case fatality rate, and that you do have patients who have fairly mild symptoms. And when that's the case, they are much more likely to be out walking around and spreading it to other people, as opposed to being confined to a hospital bed. So we know the pegs, we know the holes, and we know how serious these three guys are. We knew nothing about this guy as of last January, and we had to extrapolate a lot of what we learned from SARS and MERS. Okay. So I do just want to take a minute before we get into this idea of what we learned and how geospatial science has been super helpful, to talk a little bit about the field of emerging infectious diseases, because this has been my field of expertise since about 2005. And in those 15 years, we've seen, well, just before it, we've seen SARS emerge. We've since seen H1N1 influenza in 2009. We've seen Zika, and now we've seen we've seen MERS, we've seen Zika, and now we've seen COVID-19. And I've been doing this long enough that there are certain themes that come out that are really worth sharing to a mixed scientific audience. Every talk I've ever been to when we're describing a new infectious agent has some stupid slide with icebergs and some point about the tip of the iceberg, right? And it's a cliche, but it's incredibly true in this instance, that when in the first days and months of an epidemic, you see this and you know this is down here, but you have no idea what it looks like. And so there's always this very chaotic scramble at the beginning to learn as much as we can. The other thing that's worth pointing out is that this is more than just an intellectual exercise, right? This is set against the backdrop of very sick people and very frightened people who need answers and who need some sort of solution. So I think that the best way of thinking about this is thinking about a house that's on fire. and you have a sense that water is probably going to be helpful, but there's also a chance it might do bad things, because sometimes spraying water onto a certain type of fire is the worst thing you can do. And also hoses haven't been invented yet. So it's a very kind of chaotic feel where you're having to take best guesses based on very limited information and come up with solutions. It's an exciting line of work, but it certainly can be quite chaotic. And this is one of those moments in history where we're all of my colleagues and I are feeling very tired and just in a lot of chaos. I wanted to share 4 examples from 3 different EID epidemics in recent memory that I think share how, oops, sorry, I advanced that too quickly, share how we can use atypical techniques that you wouldn't necessarily think would be relevant in order to make practical advances toward solving an emerging disease problem. So the first image I have here is from a paper that was published in 1984. And this is, in my universe, this is actually a very famous picture. This is a paper that was about what we now know and now call acquired immune deficiency syndrome, or AIDS. In the early 80s, this was abject chaos, right? Because it was very clear that there was some type of syndrome that, at least in the US, was spreading most frequently among gay men, and it was starting to spread among IV drug users, and it was not clear what was causing it. It wasn't even clear that it was infectious, but what was done in the absence of an information, because no one had ever cultured the virus we now know as HIV, no one had seen it under a microscope, no one had detected antibodies to it, no one was sure that it existed. But what was clear is that some disease was spreading. And so this picture here is the result of an extensive amount of medical sociology interviews that took place all over the country by one team from the Centers for Disease Control. The scientists in question were named David Auerbach and Bill Darrow, and they tracked down all of these men who were sick with this apparent syndrome that, again, had no name, had no test, and they figured out who were sexual contacts of who and how this linkage went together. And then they figured out that all of this network was connected by this single individual. And so what they were able to do then was make disease control recommendations that this syndrome is apparently spread by sexual contact, and they were able to talk to people about how to stop the spread before they knew what caused it or what it was. All of this by conducting interviews. And since they had an inkling that this was sexually transmitted, but they weren't positive, they were asking about everything under the sun. They just asked enormous spreads of questions, and this is what they figured out just by doing person-to-person interviews, which you wouldn't necessarily think of. This picture over here is of an apartment building in Hong Kong. This is an apartment called the Amoy Gardens, and this was the site of an intensive spread of early SARS cases in 2003. And the oddity about this particular apartment building was that it was visited by a person who we now know was sick. He left on an airplane to Singapore and developed symptoms, had to be diverted, and then he died at a hospital in Vietnam on the way. But the reason this apartment building is important is that he visited a cousin here, and he used the bathroom, and then he left. The cousin then got sick. But SARS being a respiratory virus, usually if you see it spreading throughout a building, what you'll see is that it spreads from this apartment to this one, to this one, to this one, and down a hallway kind of sideways, because that's how the airflow is going to go. But what was odd was that in this apartment, it spread from this patient here all the way down to the bottom. It spread vertically instead of horizontally, which was odd until the engineers figured out that the sewer pipes were on the outside of the building and they were leaking. And so what was happening was that you had sewage being exposed through the windows to all the apartments below the original patients. And that's how we learned that SARS coronavirus can be spread and can be shed through the fecal tract and spread via either fecal inhalation or fecal oral contact. That was not something that was expected, because this, we saw this as a respiratory infection, but it was figured out very quickly on that this can actually be spread through the fecal tract as well. And we now know, of course, that this is true for SARS-CoV-2 as well, which is why so many places are screening their wastewater for it, including my university, which is actually done in my lab. I don't know how I won that prize, but anyway, it's interesting in there these days. So the last two things I want to show you are from the Zika virus epidemic. The thing about Zika virus that I think people don't necessarily appreciate is that it wasn't new in 2015. It was an old virus that was discovered in the 40s. It's just that no one cared about it until 2015. And the reason for that is it was associated with this very mild febrile illness. People who got it, they got fevers, they got achy joints, and that was about it. Then they recovered and never thought about it again. Starting in 2015, we saw this pop up in the Western Hemisphere for the first time. And the reason we started becoming interested in it and started worrying about it was that it seemed to be far more aggressive a disease all of a sudden in two different ways. The first way was that some clever person recognized as these Zika cases started increasing, so did cases of something called Guillain-Barré syndrome, which is a central nervous system impact. So by looking at these two peaks temporally, using pure epidemiology, a lot of physicians suddenly realized that this is impacting the central nervous system. And then they started looking for it there. And then they realized it could cause encephalitis as well. No one thought that before. The only reason anyone looked and checked was because some clever epidemiologists overlaid these two curves and realized how nicely they lined up. The other thing, of course, about Zika that was very new in 2015 was that we realized it can cause microcephaly in infants if pregnant women are infected. And that was due solely to the observation of two obstetricians who were both women in the province of Pernambuco. What they realized was they had this enormous spike in microcephalic infants being born to their patients. And they called the Minister of Health and said, I think these two things are related. The Minister of Health originally laughed at them, but they kept calling, and then they called the WHO and PAHO, which is a Pan American Health Organization. And it turns out, of course, we now know in hindsight that they were right, that those two things absolutely were related. And so every so often, it just takes somebody making a weird observation and saying, I don't have evidence for this, and I wouldn't have predicted it, but there's something funny happening here, and we should look at that pattern in a scientific way. So with that being said, I think it was important to give that backdrop to and that context to SARS-CoV-2, because it's important to think about that context when we look at what we know and when we learned it and how we learned it. So what we knew in the winter of 2020 about SARS-CoV-2 was the following. We knew what it sticks to, right? Because we knew it stuck to ACE2, because right away, once we figured out that this was a beta coronavirus and we figured out how close a relative it was to SARS-CoV, instantly that experiment was done. So we knew what it sticks to. We knew what it does. And again, this is winter of 2020. We know a lot more now. We know it caused fever and cough, something called acute respiratory distress syndrome. We're starting to have a feeling that it might cause cardiac disease as well. We were starting to notice that some people seem to have GI symptoms. And we also knew who it hit the hardest. We could tell it seemed to have a predilection for older adults. We could tell it absolutely hammered diabetics, and it hammered cardiac patients. And so at that point, again, we're looking at a house that's on fire, and we have to figure out what to do about it in the absence of hoses. At that point, we ask the question, what can we do with that information? And what we can do with that information is ask certain questions that jump right out. And the first thing that people thought about as a panic question was, are there any interactions with medications called ACE inhibitors, which are I'll talk more about what those are in a second, which are widely used, particularly in these populations. And if there is an impact of these medicines on this disease in these type of patients, we need to know that, and we need to understand what it is, like yesterday. We could also tell from this predilection that there seems to be some link to over-inflammation. So a question became apparent is, what would happen if there was widespread anti-inflammatory drug use? Would that be helpful, or would it dampen people's immunity so that they were more likely to be infected? We didn't know the answer to that. And then finally, because hoses have not yet been invented, can we use buckets instead to put the fire out? And the analogy there would be something called drug repurposing. Is there an existing medication that might be helpful in combating this virus? Okay, so with that backdrop, I'm going to tell you a good story, and I'm going to tell you a bad story that uses geospatial analysis. And those are the last couple things I'm gonna talk about. So I'm gonna start with the good story, and it relates to ACE inhibitors. ACE inhibitors are medication that patients take if they have high blood pressure. And just as a really quick backdrop, how this works is as follows. If you pretend this is a blood vessel for a minute, and this is what it would look like normally, blood would flow at a nice even pressure through here. This molecule right here, called angiotensin, this causes something called vasoconstriction. So when you have a lot of this present, this vessel clenches up together, and it becomes more narrow. That's going to increase the pressure on this side of the current if this becomes more narrow. When that happens inappropriately, you wind up with high blood pressure over here, okay? So what blood pressure medications do, what ACE inhibitors do, is they block this molecule, so they make it the blood vessel widen back up, and it takes this high pressure away. Right, so that's how those medications work. That's a good thing if you have pathologically high blood pressure. However, there was a concern when it came to this disease, because when you block angiotensin II, that increases ACE2. And if that sounds familiar, that's because ACE2 is the hole for SARS-CoV-2. So instantly there was this question, if you're taking this medicine, which blocks this, which makes more of this, does that make you more susceptible to contracting COVID-19? And will your disease be more severe because you have more holes for more virus to fit into? So the question instantly became, sorry about that. The question instantly became, should patients stop taking blood pressure medicine? Because is contracting severe COVID-19 suddenly a bigger risk than having slightly high blood pressure? That was a question we needed to answer right away. And the very first paper that came out on this happens to be a geospatial analysis. So here's this paper that came out from the Oregon Health Sciences University, and here's the link to it on PubMed. So what this group did was they looked at ACE inhibitor use by county in the United States. All of my animations went too fast, and they showed the darker blue a county is, the more ACE inhibitor use per capita there is. What they then did was they looked at COVID-19 cases and COVID-19 deaths, and this analysis was done in June. So this is data as of June. We now know these maps are going to look a little bit different today than they did then. But what analysis they ran then was trying to correlate geographically these things. And of course, they adjusted for things like population density and percent of the population who had health coverage, both to supply their blood pressure medication, but also to get tested for COVID and get care for COVID. It was adjusted by age. It was adjusted by non-Caucasian population density because patients of color have health disparities, both for high blood pressure and for severe COVID-19. And it was also adjusted by air quality, which at the time we weren't quite sure whether poor air quality would contribute to more severe COVID. And what they found was that ACE inhibitor use did not correlate geographically with increased COVID-19 cases or with deaths at all. And my first thought looking at that was that I was concerned about the timing of it, in that we had COVID first striking the Pacific Northwest and the Northeast very heavily, and so was there a temporal effect of that? But when you actually look at the earliest cases and where they were, and the ACE inhibitor use, you can see that, for example, now there's moderate use in Arizona, and a month ago this was extraordinarily just dark indigo blue, when presumably this is constant. And so this conclusion really does seem to hold up, that ACE inhibitor use really does not seem to predict COVID-19 cases or death. And that was incredibly good news, because patients were starting to stop taking their blood pressure medication, and we now know they do not need to incur that risk, and they can keep taking their medication without creating a further risk for themselves. So that was done without a clinical trial, without anything, without doing animal studies or anything else. That was done purely geographically. and it seems to have held up. So now I'm going to tell you a bad story, and that involves repurposing of antimalarial chloroquine drugs. And the origins of this story are incredibly rational, and they start with this paper that was published in 2005, wherein a group studied the impact of the drug chloroquine on SARS coronavirus replication. And if you look at just the title of this paper, it seems like chloroquine is an excellent medication that would stop the spread of SARS. But unfortunately, if you actually read the paper, this was done entirely in a cell culture dish. And when they say stop the spread, what they mean is spread from one cell to another, not one patient to another. So chloroquine and its derivatives, including the oft-mentioned hydroxychloroquine, they are very potent inhibitors of coronavirus in a laboratory. That is indisputably true. But what happened was, looking in a laboratory then caused people to start looking geographically, and images like this started popping up online, because at this point, this was now in the popular news media. So you started to see images like this. And on the left, you have an image of the malaria belt. And then on the right, you have, as of April 2020, you have the countries that were having the highest density of COVID-19 cases. And if you look quickly, they look like they're inverse to each other, right? And so this tempted a conclusion, which a lot of people made very quickly, about anti-malarial drugs and SARS-CoV-2. In other words, when you look at what's possible in a laboratory, and when you look at this kind of geographic coincidence, it's tempting to speculate that, well, perhaps there aren't cases here because people are taking anti-malarial drugs, and that must be protecting them. Of course, what we know now at this point was that there's an absolute lack of efficacy for chloroquine and hydroxychloroquine in human COVID-19 disease. And in fact, there's evidence that it's actively harmful by causing arrhythmias in patients who are already having cardiac troubles. So it begs the question, why doesn't it work? Given the culture results, I mean, shouldn't this be a good drug? Well, it doesn't work for various pharmacological reasons. A lot of things you can put on a microbe in a laboratory that might kill them are not appropriate for human use in many different ways. For example, ethyl alcohol also kills SARS-CoV-2, but you don't want to drink 70% ethanol because you will be blind, and that would be bad. But on a more specific level, we have to think about things like, well, how are these drugs metabolized? How are they absorbed? Antimalarials are excellent at being absorbed and going directly into the bloodstream. But if most of the virus replication is happening on the surface of the lungs, being shuttled straight into the bloodstream is not particularly helpful, right? The drug's not getting where it needs to go. So it doesn't work particularly well in a live human for pharmacological reasons. But why doesn't that work given the curious geographical pattern? And of course, being GIS users and familiar with this sort of thing, I don't really need to tell you all the dangers of just looking at two maps and saying that must be the answer. But in particular, there are two very clear-cut microbiological reasons why, at least I can say, that doesn't hold up. And the first is by looking at yet another map. And what this map is it's looking at the spread of chloroquine-resistant strains, which started here in Southeast Asia in the late 50s and has since spread all over the world. So one quick thing I can share is that while the malaria belt looked like the inverse of early COVID-19 patterns, chloroquine and hydroxychloroquine are not very frequently used anymore because there's widespread drug resistance. They simply don't work very well to treat malaria anymore. So all of these people that we might have assumed were taking these drugs really aren't taking these drugs. They're taking other antimalarials. And then the other microbiological reason I can share with you has to do with this organization right here. So this is a program, LabCap, that's run through the American Society for Microbiology. So I've been a member of this since 2000, and I've been a member of this since 2008. So what we do within LabCap is that the mission is to build international laboratory capacity. So we go to different countries and we try to figure out what instruments are needed, what are practical realities given things like the infrastructure of a given clinic or hospital, including things like what is the power grid like, what is the refrigeration situation, what is the storage situation, and then we go and we train local technicians to do diagnostics in a way that is needed and appropriate and feasible and sustainable in various LMICs. And that being said, looking at that other map, the way that we were diagnosing COVID-19 was using something called quantitative PCR. Very few countries in the malaria belt really had the infrastructure in place to routinely diagnose COVID-19. And so I looked at that and just said, I think your absence of cases may be absence of detection, point A, and point B, that drug is not used in that area due to drug resistance. So really that map looked tempting, and I think people without knowing various microbiological caveats and certainly geographic caveats that I'm not aware of, it looked rational to people, but it just turned out that that was not the explanation. Okay, so in summary, what we've talked about is that SARS-CoV-2 is undoubtedly a zoonotic spillover event, and we are 99.99999% sure its reservoir species are bats, and its secondary host we are still trying to figure out, although the mustelids are looking very promising. COVID-19 is a new disease that's at pandemic proportions right now, and that many sciences, including the geospatial sciences, have contributed substantially to our understanding of this disease and what to do about it. So with that, thank you very much, and I'd be happy to take any questions and things back to Patrick if
Sure.
That is
yes,
what I'm supposed to do.
Thank you so much. That was very interesting, and obviously this topic raises so many questions. I think you certainly introduced some different thinking around, different than what I think a lot of geospatial users are talking about. One thing we did have conversations with on the MEGUG board is related to proper geospatial map display, labeling, sourcing of data, and being aware that we have to provide all the context for the maps that we make, and some of those maps that the malaria inverse map, if you will, is one of those kind of scary maps that without the right context kind of makes GIS look bad.
Absolutely.
Yeah, GIS professionals are ones that we really believe in the data and what the data tells us, and then we try to present a visualization of the data to help people make decisions, spatially. So do we have any questions? I can see some coming in here. I just need to open the questions panel all the way. See if I can read this one. We've got some. Jeff, maybe while I'm doing this, I don't know if you have any questions for Meghan.
Sure. Yeah, so building on what Patrick was saying, when you put up those two maps next to each other, I think one of the first things that popped into my mind was, I'm sure all of us on the call and the seminar understand the importance of knowing your data well and knowing what it is that you're presenting. And if you're given a data set that you don't understand, it's very easy to display results or imply results that might not really be there. And one of the first things I thought of when I saw that map of the malaria belt was, I don't know the medical side of it. I don't know how people's treatment or care works, but my first thought with my limited experience in one of those areas was that just because a place has a lot of malaria doesn't mean it has a lot of people who are prophylactically on anti-malarial medications.
Exactly.
It may very well be that the only thing that would even imply would be that if a person already had malaria and was taking medication to help them get over the malaria, that they may be less likely to get COVID. And that's ignoring everything else you said about whether they're even using chloroquine derivatives or not.
Exactly. And I have to say, just to hopefully redeem that slide a little bit, is I tried intentionally to find an image that was from a news story that was talking about this potential, use of this drug, because it was so not scientifically drawn.
Right.
And so I went back and forth about that, thinking like there are probably much more appropriate looking maps that one could do, but I think the reason I wanted to show it as such was because I think so many people saw that image of those two maps put next to each other, and they didn't have either the geographical or the medical context to be able to apply kind of scrutiny to it. And all they saw were two maps that looked very much like they were inverse to each other. Right. And so it really created a lot of challenges when we're trying to do communication and outreach to people in terms of, well, no, I know why people thought that might be a good medication, but it turns out it's not. And yes, I know that map kind of makes some intuitive sense, maybe if you don't have the context to look at it properly. But it's hard because I think it inserted a conclusion into a lot of people's heads without proper vetting from either expertise.
Yeah, which is unfortunately a very common thing, it seems, these days. We do have a question on, let's see if I can get this right. Are there any studies, or is it just your general sense on zoonotic spillover? Are these events fairly regular, somewhat evenly spaced in time, or are they becoming more or less frequent?
That's a great question. So they are undoubtedly becoming more frequent, and I think a lot of that plays into things like habitat use and encroachment. And we are far more connected as a, I know that's a cliche, but it's absolutely true. I look how fast this disease, even compared to SARS 17, 18 years ago, look how fast this spread all over the world. When the transmission is essentially the same as SARS-CoV was. There are far more international flights, so even when there's a zoonotic spillover event that can lead to person-to-person transmission, it tends to go faster and become larger. There are more spillovers in and of themselves, again, because we are just encroaching so much on habitat, and it even goes into things like things that aren't necessarily a classic spillover disease, but certainly have zoonotic overtones to them. Something locally we think about is Lyme disease. We have far more of that now than we used to, and a big part of that has to do with habitat destruction, right? So if you have wide expanses of forest versus small pieces of it broken up, you tend to have greater instances of deer, which are amplifying hosts that are walking in and out of people's yards. You tend to have more mice, which spread exotic ticks farther and to more people, and kind of serve to shuttle the ticks between the deer and the people. And you have fewer mouse predators that you might see in a deep forest when you have these little patches, right? So there are fewer owls in a teeny patch of forest than in a big patch of forest. And so even something like that is our habitat encroachment has really forced a lot of interactions with wildlife and removed a lot of wildlife interactions that were really keeping these things in check. So it's very much a problem that's increasing in frequency. It's not just our ability to detect it.
Well, it's interesting because what that sort of implies, and maybe I'm just reaching with this, but is how much science plays a role in understanding things from biology with climate change. And if we have unfettered climate change and we have changes in habitat like what we're seeing, these things can all tie into these sort of pandemic situations.
Yeah, no, no, you're absolutely right. And there's an enormous amount of energy around that exact problem, because if you look at climate change and changing habitats, there are several avenues to that, and the first being an obvious one that jumps out is things like vector ranges, right? So you may have mosquitoes and ticks that are local to an area that they didn't used to be local to, right? Because the climate is now favorable for them to live there, where before it might have been too dry or too cold or something like that. So vectors are an obvious one, but another is that, and this is something I think people don't think about as much, but it became very important during the influenza pandemic, is the changing climate has changed bird migration patterns, and that you have birds overlapping in, say, in the islands off of Alaska that didn't used to overlap with each other. And so that's a bridge between the Eastern Hemisphere and the Western Hemisphere that did not used to exist. And so when you have things like influenza strains that kind of cook and simmer in wild birds, you can have a transmission from one hemisphere to the other that you wouldn't have had 20 years ago. And so that is absolutely not a reach at all. That's something that it keeps a lot of us up at night, actually. So
yeah. Well, we're running a little bit over. We do have just one last question that we'll ask. It's certainly there's a lot of questions I think on everyone's mind, but it would be interesting to hear if you have any insight into geospatial science or relation to contact tracing, or the current state of how scientists are working with that, or.
So I do know that it is being used, but I am not super familiar with it, to be honest. And actually, Jeff and I were talking yesterday about something of a related element that there's a contract tracing app on iPhones now, and if you activate it, it can it can follow you and tell you who not individuals, but which iPhones were near which other iPhones in the past 2 weeks to aid in contact tracing. And you could certainly see where that would be almost instantly mappable. And so this is happening on a governmental level, but I think there are also companies that are trying to create tools for doing this as well. Sure.
Yeah, there are, and I know that in other countries they're adopted much more widely than maybe here in the United States. It certainly, that would be a great topic for one whole webinar to hear more about how that is actually being done. I know that the state is doing much more and more of it every week, it seems. But that's fine. That's great. We'd like to thank you so much again for your time, Dr. May. It was very interesting, and it was interesting to see more of the biological science of things than we, I think, have been all exposed to. So it's good to round out our understanding of the nature of disease like this. So thank you so much for your time. Thank you, everybody, for your time today. We will be posting the recording of the webinar in probably a week's time or so. Thank you, Jeff, for your assistance.
Thank you, and thank you, Megan.
Thanks for having me. It was a lot of fun. Thanks.
Excellent. Thanks so much. Have a great rest of the day, and we hope to see you on next month's webinar.